will start off and introduce myself. I am Cindy Haba, and I'm the host for tonight. I'm a PNRI board trustee. I'm the chair of the development committee, and as the founder of Momentum Coaching, I'm also tonight's sponsor for this event. Momentum Coaching, we are a coaching company working with high achieving accomplished professionals in the areas of executive coaching, career growth, and career transition coaching and small business coaching. So we're happy to sponsor our very first science and wine event. And so you guys are all part of our new event series, and we're very excited to kick this off. As you probably know, as we know, science has become pretty important in this last year, right? I think we all are talking about it a lot more. And it's one of the primary conversations that are happening in all aspects of our lives. And which is very energizing and exciting for PNRI. We want to support those conversations and encourage those conversations and fuel that interest. We want to be part of the conversations that happen about science. And in fact, PNRI has a very long history of pairing the stories of science and the wonder of wine. And so our Science and Wine Club is a natural new series for us to start off with. And so we're very happy to be starting this new program together with all of you. At PNRI, we make a very conscious effort to put our science first, and which is why we call this club our Science and Wine Club, rather vice versa. Our amazing researchers and scientists are at the center of everything that we do. And we feel that it's important and necessary and fun to hear directly from them, straight from the source, to hear what's going on in the world of science. And so to that end, Rick McLaughlin, one of our PNRI researchers, is going to talk with us first this evening. But before I launch into Rick and let him tell us some of the amazing things that he does, I just want to share with you some protocol and how our evening is going to go. Nobody wants you to wait to start tasting your wine. I see some of you already have glasses in hand, so I'm glad for that. So please go ahead, open your bottles, enjoy your wine, and go ahead and be sipping right along as we talk. We will start off with Rick, and then we're going to ask Denise and Brett Isenhower to join us and do the wine tasting. But don't wait. Go ahead and enjoy some of that now. And then immediately following both of their talks, we are going to have time for some Q&A. So I'm going to ask you to come up with questions for our speakers this evening, but hold off on asking them until our Q&A session, and we'll be able to get everyone's questions answered. And so, as we were planning our Science and Wine Club event series, we wanted to come up with a theme for our evening. And tonight's theme is evolve. And so evolve. So what does that mean? How did we come up with that? Well, Rick, as a researcher at PNRI, has a lifelong passion for studying the evolution of genes and the genome. And coincidentally, Denise and Brett, winemakers and scientists themselves, actually, they know a lot about cloning and the evolution of grapes. So it just made sense to talk about evolution and to bring these two topics together for a perfect pairing that we think. Evolving science, evolving wine, evolving together. And so, did we have the results of our poll, Amy? Can we share that so we can see where folks are coming from? Thank you. Fantastic. So we have a lot of friends that invited you tonight, which we love to see that. And family members, thank you, all you family members that are here tonight. I think Rick's mom is on the call, and I think my mom's on the call. So yay! Let's hear it from mom.
Hello.
Fantastic. And so this is great. We're glad that you are all here. We're glad that you have found us, and we know that you're going to be in for a treat tonight as we learn about some evolving science and evolving wine.
And can I interrupt just for a second to say that the farthest away person, I think, is Florida, unless I missed someone farther away. And I didn't see a huge number of like a room that's the biggest room, but I did see a lot of +1 dogs and +1 cats, which, you know.
That's very important. Very important. We like that. And we're glad they're here. All of them, 4-legged and 2-legged alike. Good. Thank you, Becky. And so as a board member of PNRI, one of my favorite benefits is that I get to hear our scientists talk about their work a lot. It's a really nice perk. And so I learn all sorts of fascinating things every time we have meetings. And I know how much they love to share their research. And in fact, when Rick heard about our new Science and Wine Club event, he was the first one that jumped at the opportunity to speak tonight. So we were very thrilled that he was as excited as we are. I'd like to introduce Rick. Rick and his lab joined PNRI in 2018, and his research is complex. His research is leading edge, and he always manages to make it fascinating and understandable to scientists and non-scientists alike. So it is my pleasure to introduce you to Dr. Rick McLaughlin. Rick, I'll let you take it away.
Thanks, Cindy. Thanks a lot. Thank you for sponsoring this. Thanks to Momentum. Thanks to Isenhower for sending the wine. I got it for free, so that's pretty cool. And I'm super excited to be here. So let me just get these slides up and going. All right, does that look good to everyone? Okay, so thanks again. Try to keep my talk to 10 minutes. If you know a scientist, you know it's really hard to only talk 10 minutes. So like this took me a lot more practice than an hour-long talk, but I'm really excited to be here today. So I'm going to tell you a story today that to me is like one of the coolest things that I've ever learned about in biology. And at the end, I'll bring a little bit back around to what we're doing in my lab, what we're doing at PNRI. But I just think this is like such a relevant and contrasting story to the things that are happening in the world around us right now. So hopefully you all find it pretty cool also. So I have this picture of, called Tree of Life that's drawn by this guy named Charlie Harper. And I always show this picture because the reason I got into science, I think, was really that I was just obsessed with nature. I'd be outside checking out all these things. And I think it's obvious to all of us that this like amazing diversity of form and function that exists in life is, it's just overwhelmingly beautiful in a sense. And, the last 200 years or so of evolutionary biology has really shown us that, all of these amazing forms and function, these colors, these shapes that exist in humans, that exist in nature around us, they all came from this series of modifications, which happened gradually, but some of them resulted in these very dramatic advantages and changes to the forms that we see around us today. So my lab really capitalizes on this to try to understand how it is that the human body functions, how it is that we work, and ultimately like how it is that our genetics, our history gives rise to how we work. And in the context of disease, what's breaking down there. So the idea is basically if we understand where we came from, then we better understand how we work. And biological systems, organisms, plants, trees, humans, just like we as individuals are shaped by what's happened in their past. So, our lives have been shaped by our family, by our ancestors before that, and we are who we are because of those experiences. And biology is the same way. So here I'm showing you this tree of some organisms. But, the basic idea is if you take two organisms like that, this baby gibbon and this baby human, which is my kid who's now 5, which is kind of hard to believe. We know that about 18 million years ago, these organisms shared a common ancestor. So at that point, they were basically the same thing. And in those intervening 18 million years, it's all these changes that have happened in the genome of these organisms, the code that encodes all the information necessary to make these organisms what they are. All those changes resulted in these dramatic differences that we see outwardly, and these dramatic differences that we see inside the genomes of these organisms, the untold differences in the mechanisms by which these organisms function. So, basically we like this quote from Shakespeare, what's past is prologue. What's happened in the past gives rise to what we are today. And by understanding what's happened in the past, then we can better understand how these things work, how these things function in the present day. Okay, so I think this is no surprise to anybody that one of the major drivers of all these changes that have happened basically to every single life form on the entire Earth is viruses, right? And this is a topic that is all too familiar to all of us right now. But it's very clear that just like we're witnessing in the world around us right now, this over and over repetitive infection of our species by viruses has been happening for basically since the existence of the very first life form. So, this idea is all familiar to us. We might have a host like a human. In a good situation, our immune system blocks the ability of that virus to replicate. But as we see outside right now, that's not always the case, right? Sometimes there are viruses that are new to a species, or viruses that change in a way that get around these mechanisms that exist in our genome to block these viruses. And that's bad. In that case, the virus replicates, and the disease that's associated with that viral replication obviously impacts us as humans, impacts other species too. But what's really interesting is that over long time scales, not tens, hundreds of years, but tens and hundreds of millions of years, there's this back and forth between the states. There's this evolutionary arms race, as we call it, where organisms are always fighting against pathogens like viruses to block them, to do better, to not die. And viruses are always changing in ways to try to get around those mechanisms by which the host uses to block them, trying to replicate themselves, trying to survive themselves. So, we all know viruses have done bad things, but what's really amazing and what's really testament to this huge impact that these viruses have had on the evolution of all living creatures is if we look inside our genome, right? So our genome is about 3 billion base pairs of DNA. That's 9 zeros. And if you imagine this gray pie chart here representing those 3 billion base pairs of DNA, what's really impressive is if we think about our form, our function, the way in which the human body works, the sort of workhorses that make that the way it is are proteins. So you might think, all right, if you're going to write a piece of code, you're going to write an instruction manual, which is kind of what the genome is. You really want that to be information dense. You want it to be rich with information, with instruction, with things that are important. So you might guess, all right, this genome is mostly the information that's needed to make those proteins that create the form and function of our body. But what's really fascinating is that only 1.5% of all of those 3 billion base pairs in our DNA are needed to encode the 20,000 or so proteins that we need to make a human or a higher organism like a mammal. What is to me even more striking is that this huge impact of viruses, which again, we're seeing right now and has happened for millions and millions of years, is actually really evident in our genome. So what's been found is that 8% of our genome has actually been created from DNA from these viruses. So, our genome is a record of these over and over assaults that have happened from these viruses over evolutionary time. And you might ask, all right, how do you get virus sequence in your genome? But what's going on here? It's really weird. And much of the sequence actually comes from this particular kind of virus called retroviruses. And all you need to know about retroviruses is that they're a virus that oftentimes has an envelope around it, so a container in a sense that holds the genetic material, the instructions for that virus to do its job. And what happens is that virus comes in contact with our cell, it fuses with our cell, and it releases its contents inside of our cells. So what's special about these retroviruses is they go into the nucleus of our cells and they actually stick their DNA, they cut our genome open and insert their DNA into our genome. And it's this process that's happening all the time when we experience infectious retroviruses in our environment. But what's really interesting is that sometimes these insertions result in the virus making more and more virus, but we're kind of stuck with that insertion, especially when these viruses insert into special cells called germline cells, like sperm and egg. So in that case, these viruses are actually passed on to our offspring. So that leads to this bloat of the genome, but it also comes with this other side. And that is that if by chance that chunk of virus DNA in our genome can become useful to us, maybe our genome will figure out a way to make use of it. And we call this process domestication. So this is basically a genome taking a piece of virus DNA, stealing it, and making it its own. So this process of domestication has had some huge impacts on the evolution of our species. So here I'm showing you a tree that shows the evolutionary relationship of some species. And one thing that's really interesting about mammals compared to all these other vertebrates is that all these other vertebrates lay eggs. Mammals don't lay eggs. And actually, if you look more closely at mammals, there actually are some mammals, as probably we all know, that do lay eggs, like these monotremes, like echidna or duck-billed platypus. And there are these other class of mammals called marsupials, like opossums and koalas, that keep their offspring in a pouch in the front. But the big difference that emerged after the split of these organisms was the development of a placenta. A placenta is kind of amazing. It's actually this barrier whereby you can raise your baby inside of you for quite a while and protect it from all the bad things in the world, right? So here's sorry for the complicated diagram, but basically what this placenta does is function as a barrier to separate mom's blood from the baby. And why would you want to do that? Well, that's because mom has antibodies that could attack the baby. This is basically, a parasite in a sense that's growing inside of a mother. This is a foreign organism. And sometimes there's incompatibilities there that can cause problems. But really, you want to stop viruses, you want to stop bacteria, you want to stop other pathogens from making their way to that developing fetus. And that's really a hard problem. So, viruses, as I showed you, they're really good at binding to cells, fusing, releasing their contents. They're really good at sneaking through tiny, tiny spaces between cells. And the placenta is really possible because of an amazing contribution from a virus. So, as I showed you, viruses are really good at fusing membranes together. And it turns out that when these placental mammals evolve, that corresponds perfectly with this innovation in the genome where this mammalian ancestor seemed to steal a piece of a virus, the viral envelope, to create this new gene called syncytin. So what is pretty amazing is that that placental organ is actually comprised of this huge layer of cells that have all been fused together into this sort of balloon that's a double layered organ that blocks the outside of that fetal environment from all the bad things that are happening. And that probably never could have happened without stealing this essential function of viruses fusing together to make this placenta together. So in a sense, what I'm telling you is like, we are placental mammals. Our biology is the way it is because of the presence of these viruses in our organism. So just to wrap up, this is not a unique phenomenon. This has happened many, many times. And one thing our lab studies is not how these domestications of pieces of viruses give rise to new developmental phenomena like placenta, but actually what we know is sometimes these domestications happen and they're actually used as a weapon to block new virus infections that an organism might encounter in their environment. So sometimes these things look a lot like viruses. And when a new virus tries to come in, the presence of that old piece of a virus is actually a really effective way to block the invasion, the infection by these new viruses. So this is something our lab is really interested in studying. We're thinking about this in a lot of different ways. And while the viral defense thing is really cool, this idea that this amazing fundamental part of mammalian biology has really been shaped by the presence and the co-option, the domestication, the stealing of this piece of virus is, to me, it's just really fascinating. So I'll end there and I'll give a big thanks to my team. PNRI as a whole has been a great place to start a lab. Again, a special thanks to Momentum and Cindy for sponsoring this, and a huge thanks to Isenhower for providing the awesome wine. So with that, I will stop, and I think we'll move on to wine, and then after that, I'll be super happy to take your questions about the science. Thank you.
All right, so I'm Denise Isenhower. This is my husband, Brett. Oh, yeah, thank you, Rick. But
soft applause.
Soft applause. So Brett and I, we, as Becky had said, or somebody had said, we are pharmacists. We met actually at a hospital pharmacy in Indiana where we both grew up. We proceeded to get married and leave Indiana so Brett could pursue his MBA. While we were there, we were kind of making beer and wine for fun. Trying all kinds of things, doing different types of fermentations, and decided we liked the wine business over the wine or the beer business. And we started looking all over where we wanted to move to and make wine. And Washington was kind of on the radar. And so we actually fell in love with Walla Walla. We could both get jobs here as pharmacists. It's a beautiful small town. And so we moved here in Walla Walla in 1998 and started the winery. So we are starting our 22nd year here in the wine business. It's been a very interesting 22 years. We do have our winery here in Walla Walla. So those of you who live in Washington, we'd love to see you come visit us once things open up. Well, we are at 25% now, but, and then of course we do have a tasting room in Woodinville as well. We make a little bit of everything just because we like to keep things exciting and fresh and fun. And so the first wine, since I know you guys did start drinking a little bit earlier, is our Road Less Traveled. For those of you who don't know, Road Less Traveled is actually a poem from Robert Frost. It's one of Brett's favorite poems.
Two roads diverge in a wood, and I took the road less traveled by. And that has made all the difference.
So our goal in life is to be a little bit different. The one thing about Cabernet Franc is it's not, well, when we first did this wine back in 2002, it wasn't the most common wine that you would see out there, particularly as 100% varietal wine. And so this is a 100% Cabernet Franc. And for those of you who don't have our wine, Brett's going to tell you a little bit more about Cabernet Franc and where it came from and all that, because we are on the Evolve thing tonight. But some of you did get our tasting notes. And basically, for those who do have our Cabernet Franc tonight, this is a blend of two different clones of Cabernet.
It's one clone.
Oh,
Two
vineyards,
two
one
vineyards,
clone.
one clone.
Sorry. Excuse me. Anyway, so I will let Brett talk about the clones and the wine because that's kind of more his passion, where I get to do all the boring stuff with compliance and shipping wine and all the other stuff. So
Yeah,
further ado.
It's all romance until you have to do the monthly reports for the state and federal government in the wine business. Just briefly about the Road Less Traveled. And we'll talk more about clones here in a moment. It's what we call clone 214. So it's from two vineyards, but it's the same clone. One of the beautiful things about wine grapes is they totally express where they come from and who they are better than any other agricultural product out there. And I'll kind of describe why. I think that the wine grape is one of the most complex plants that we get to enjoy as humans. And for some crazy reason, we as humans have been dragging the wine grape with us wherever we go throughout recorded history. The wine grape vine itself, its genome, it's called Vitis vinifera. The research I did saying there may be 10,000 different varieties of Vitis vinifera out there. So that is the European wine grape. But I don't know if there's actually 10,000 or not. It seemed impossible to actually verify that. I can say that humans make wine from 1,200 different grape varieties. But there are what we call the 20 international varieties of wine grapes. So when I say a variety, that would be Chardonnay, Cabernet Franc, Grenache, Malbec, Pinot Noir, and so on. I mean, all of you could probably list the 20 varieties you see in stores, you see, well, when restaurants open up again, you'll see in restaurants that you're familiar with. But every one of those varieties is Vitis vinifera. They all have the same DNA. And so the question I have for you is so how, and kind of talking about with the DNA of grapevines, is where do baby grapevines come from? I mean, if you think about it, I mean, how do you get a baby grape? We know as mammals how we get a baby or a baby mammal, or baby humans. We all are relatively aware of how that occurs. So there are multiple ways to get a new grapevine. So the one way is technically a sexual reproduction of grapevines, where grapevines will pollinate each other. Then you'll get a seed. So, and then the grapevine says, well, I think of grapevines as very feminine. They're mommies. And the grapevine seeds are the babies. And what they want to do is they want to make the berries get sweet. So you've got sugar that accumulates as we get into the summer and fall. The seeds get harder and harder from the tannins as they develop. And the grapevine's goal is to get its seeds ripe enough, or sweet enough, that a bird will fly along, take a bite of the seed, or bite of the grape, swallow it, and it'll fly off. And a mile later, it will process the sugar. It will have a little diarrhea, and it will deposit the seed somewhere on its flight path. That seed has gone through its digestive program. It lands in the soil, and magically, the next spring, as the soil starts to warm up, a new grapevine grows. Now, what's interesting is that you had a mommy and a daddy for those two grapevines, for that seed. Now, you could have a Chardonnay mommy and a Cabernet daddy. You don't know. You could have a Chardonnay mommy and a Chardonnay daddy. But that new seed is a new grapevine. There is no guarantee that 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 new seed is going to become, so let's say, a Chardonnay, because you've got mixing of DNA, and then what's going to be expressed by that seed is going to be different. And so you get new grape varieties. One way, the traditional way, is you plant seeds, but it won't be the same. It's just like our children are not clones of us. Our children are new human beings. Same way with seeds creates a new grapevine. Now you say, well, how do I get Chardonnay from Burgundy, France to somewhere in Eastern Washington or Western Oregon or California? The other way to do it is called vegetative propagation. You take dormant wood, which is the growth from the previous year.
Wood being the vine.
The vine, you cut it. Now the vine has buds. Those buds is what's going to be the future growth. Now, if you take that dormant wood, you cut it off, stick it in the ground. Then as the season warms up, it gets above 50 degrees in the springtime, that will start to grow. Those buds will actually push out new growth. Now, you notice there is no sexual reproduction there. It's vegetative growth. And so what occurs in that point is you have recreated the mother. So there is no sexual reproduction. It's vegetative. So if you want to grow Chardonnay, you want to grow Pinot Noir, you want to replicate exactly what that mother comes from, you take that cutting, stick it in the ground, and it will grow a new exact clone of that variety. So grapevines are very industrious. We figured it out as human beings, how to replicate the grapevines, how to take it with us. We could either take seeds or we could take cuttings, as we call them, and we can replicate them.
So talk about the different clones. So
what's fun about this is you get mutations within grapevines. You think about grapevines in vineyards. They sit out there full of sun. Sunlight has radiation. Radiation causes mutations. You've got the sunlight coming in, it changes, and you get cellular changes within grapevines. And within a vineyard itself, you'll have grapevines start to change. Whether so some of the times the damage from the radiation, you won't know it. You won't see it. The vine continues to grow. It produces grapes, but sometimes you will see it. Sometimes it's detrimental, but sometimes it's beneficial. And grape growers will go out and they'll survey their grapes, particularly in Europe, and they'll look at that and they'll say, well, this grapevine looks different than all the other ones. They will take cuttings, they'll take vegetative cuttings, and then they will study them. They'll try to remove the viruses from them. They'll grow them up again. And then they'll make wine from them. They'll say, okay, well, this clone or this particular grapevine makes a better clone or a better wine than another one right next door. And that's how clones occur. So when we say this Cabernet Franc, the road less traveled, is clone 214, there's an organization in France called Untitled, and I'm sure it's a beautiful sounding name, but that's the acronym. And they actually go out and survey many vineyards all along in France, collecting all these clones. They grow them up. They created this huge database of all these different grapevines. And it's kind of amazing. Some grapevines are very clonal, which means that they mutate easily. Pinot Noir is the most famous for being the most mutatable. If you think about it, Pinot Noir, Pinot Gris, and Pinot Blanc genetically are the same vine. They're mutations. They have a color mutation within the DNA. So the vine actually expresses different amounts of color inside of the grape skin. So next time you drink a Pinot Gris, realize you're really drinking a light colored Pinot Noir. Or if you drink a Pinot Blanc, you're really drinking a very, very light colored Pinot Noir. And they're the same vine. They're just mutations, they're clones. And so that's kind of the fun part of grapevines, is we've been able to analyze this. Another way that grapevines, you get new grapevines, is you have cross-pollinations. So you have a mom and a dad, they have an illicit relationship, and they have a new baby. So one of the most famous one is before the 1600s, there was no such thing as Cabernet Sauvignon. Didn't exist. The Romans didn't have Cabernet Sauvignon, wasn't around. It was a crossing between Sauvignon Blanc, which we'll call the mommy, and Cabernet Franc, which we'll call the daddy. They were crossed, they exchanged DNA, the seeds were planted, and all of a sudden, voila, that seed, a different variety is Cabernet Sauvignon. It's kind of a cool thing. So all of our grapevines are, that we know of as varieties or crossings, but then the clones are vegetative cuttings that through mutation are slightly different. And what's really fun for me as a winemaker is actually tasting the same vines from the same soil, but different clones, and seeing how different the wines are. And that's really the coolest part of winemaking, is when you see just kind of how it's like, there's nothing different. The only thing that's different is that the DNA has been changed for this one variety. And so now it's a different clone. And that's one of the cool parts about winemaking. And it's a shame you don't have an Oregon winemaker, because they love clones. It's like a Star Wars movie with them. They think it's the greatest thing in the world.
Well, and Washington is kind of newer on this clone thing. Like, we're probably maybe 10 years into the clone thing.
We are into the clone.
Some of the grapes that we buy, the farmers are like, we have no idea what daddy planted. Yeah. I mean, no clue. So, whereas now as new stuff is being put in, you can pick the clones, and we've been able to do that. And we personally don't have any of our own vineyards. We tried it and it failed miserably. So we actually buy all our grapes, and we have acre contracts with our growers. And with a lot of them, we've been able to pick the type of clone of the different grapes that we want. And so, like the Cab Franc that we're having tonight, we were able to pick the Walula Cab because we knew we liked clone 214. So.
A lot of Cabernet Francs can be very, what we call red fruit flavors and kind of herbal. Clone 214 is very blue fruit, lots of blueberry, has kind of a sea salt character, and it's very much less herbal.
Yeah. And
it's a real attractive, and you see the differences in the vines. The leaf shapes will be slightly different. The cluster sizes will be different. The tannin levels in the grapes will be different. And most importantly, the wines from these different clones taste different. And so for me as a winemaker, it's really fun to be able to kind of like mix and match, to play around. Although unfortunately, my little games of mixing and matching takes 3 to 4 years of planting a vineyard and then seeing how long it takes. And then you make the wine, and then the vines get older. And as vines get older, just like children, they get better. And so this is fun with the 2018. These are vineyards that are getting into, much older. The roots are much more established. And that's when wine starts to get fun, is when you have mature vines really showing off what they can do.
Okay, so this is a good segue for you to be quiet for a second. So we had a poll question that we put out there about what is, if anybody knows what the highest planted Cabernet, or the highest planted grapevine out there. So if anybody knows. And there's a trick one on there. Did everybody answer? Maybe, maybe not. Anyway, so this might give the answer away, but that's okay. So we're going to move on to, we were talking about the clones. And so this next wine that we're doing is actually a blend of 3 different clones. It's our 2017 Walula Vineyard Cab, and we had this vineyard planted for us and all the different clones as well.
So Cabernet Sauvignon is the most highly planted grape in the world now. And in Washington, I did the math yesterday. There's at least 14 different clones of Cabernet Sauvignon that I know of that's planted. I've made wine from 8 of those different clones. We had Walula Vineyard planted in 2014, and I picked kind of my 3 favorite clones that at the time that I kind of knew of, and I asked other winemakers what they thought. So this was a blend, and we get into weird numbers. I mean, they don't have names. They're not like your dog. So this is a blend of clone 7, clone 33, and clone 412. And basically these names just strictly come from either UC Davis, which does a lot of the clonal research in the United States, or the Ontav organization, who's in Montpellier, France. And they just assign numbers. I mean, they get all these different cuttings in from these vines, and they just assign numbers. And then they grow them up. So I know it sounds like a Star Wars movie, and it kind of is, but that's what makes it fun, especially with Disney Plus. You can watch every Star Wars movie, for 12 hours a day like our kids do when they're locked in at home during COVID shutdown. But clone 7 is an older clone. That is the Napa clone. When you taste like traditional Napa Cabernet Sauvignon, most of it's Clone 7. Our primary clone here in Washington is Clone 8. And there's not much difference between the two, you would think. They all came from the same location, but I think Clone 7 is one of the most beautiful Cabernet wines I've ever had. Clone 33 is a much newer clone that came out of Bordeaux. It's very different in terms of tannin levels. Clone 33 is much more tannic than Clone 7. Clone 7 is much more plush and lush character. Clone 3 is very tannic. It's kind of one of the hot clones here in Washington. A lot of vineyards have planted it. And then the last one is another, it's from the Medoc region of Bordeaux. It's called Clone 412. I'm a real big fan of it. Every year, it's got a super intense herbal character. And, you know,
not green, but herbal.
Not green, but herbal. It's savory. And so all these vines, this is all from, this wine here is from 2.5 acres.
Wrong one.
Wrong one, the purple label. This wine's from 2.5 acres, planted all in a loamy, silt loam soil over basalt rock on the cliffs above the Columbia River in Benton County. All the rows are right next to each other, but all the clones are completely different. And it's because the DNA is differently expressed by these vines, to which I think is one of the coolest things in winemaking. And that's why it's just, wine is such a great agricultural product because it's so different. You know, it's not like milk. You know, it's not like, it's not like anything else. Maybe coffee might be the closest to being as complex as wine. But that's what makes it so much fun, is to enjoy that all this is Vitis vinifera. It's all the same plant. It's just how we as humans have interacted with it, because we as humans really like alcohol. And so we bring it with you everywhere we go, and we like to have a lot of fun with it.
Oh, and tell them, like, I think the oldest grapevines that they found, or the oldest grapes, was like from 7,000 BC in China.
They found grapes. They found wine presses, Canaanite wine presses from 1600 BC, so before Moses.
Pretty cool.
You know, we've been doing this for a long time. In the Port region, they've been making wine for at least 2,000 years. Even the Romans revolutionized barrels. I mean, the same barrel shapes that I have now, the Romans created that shape of barrel. So it's pretty neat. I mean, it's, as I go back and forth, wine and humanity have gone together, you know, for at least 5,000 years, and we have a great time doing it, and we enjoy sharing it with you.
Okay, so at this time, I think it's time for us to be quiet and move on to the question and answer. So I'm going to mute us now.
Denise and Brett, that was fascinating. That was so interesting. And I am thoroughly enjoying both of my wines here tonight as I'm listening to you talk. So they're wonderful. So thank you. We want to jump in and do some Q&A. I'm sure that both of these conversations have prompted some very fascinating conversations from our community here. And I thought I'd start off one with Rick, jump back into Rick's talk, because Rick, that kind of blew my mind. And so I have a question for you, if you don't mind. I'll just start us off. You've told us, you know, there's some good things that viruses have done, and obviously we know that there's bad things that viruses can do. We're very clear about that these days. So overall, are viruses good or bad? How would you answer that?
Yeah, it's a fun question. It's a little bit philosophical, but you know, the way we like to think about it is making lemonade from lemons. So there are these bad things that exist around us, and you know, to question whether we would be better off with or without them is a difficult thing to say and a little bit semantic. But when posed with all of these viruses around us, when presented with all of this DNA inside of our genome, so much time has passed that if it happens to be good, you know, those benefits will be selected for. And, you know, here we are with this placenta, this immune system, this biology that's been really shaped by viruses. So it's fun to think about, and it gets funner the more wine you drink to think about. But it's an interesting question.
That is good. Yeah, I guess you're saying that time will tell, right? See if it helps us or hurts us over time.
Yeah, it's all a little bit relative. It's all kind of how you think about it. So
Yeah.
You make do with the bad things that have happened, and yeah, make lemonade out of lemonade. Make lemonade out of lemon. So
Right. Okay, our next question is for Denise and Brett. We have a question. Do all winemakers keep track of which clone numbers they use? So if we like clone number 214, would it be easy for us to track down the other wines that also use this clone? Is that a thing to do?
Actually, it's not. A lot of winemakers either don't pay that much attention to it, or they de-emphasize it in certain ways. So it's a hard thing to do. A lot of growers, maybe like some of the older vineyards out there, they may not know what was available. This kind of revolution in clones that are available now really only started in the 1990s with DNA technology and the fingerprinting and being able to figure out what is what. Before that, and it's a lot of anecdotes of, well, this is what grandpa had. Or, well, I bought this at one time. My dad, who's passed away, knew what it was, but he didn't keep records. So we hear that a lot, even from vines that were planted in the late 70s and early 80s. The genetic diversity of grapevines was, back for the earliest plantings, say in Washington in the 1970s and 80s, they had one Cabernet Franc. They had one Cabernet Sauvignon. That was it. For a long time, there was one Syrah clone. That was it. And now we've had this explosion. If you go back to the earliest plantings in California, it was Italian immigrants who basically put sticks in their pants, got on the boat, came over and stuck them in the ground in Napa and Sonoma. I mean, and that's why you see these kind of these field blends, Zinfandel wines that are in Sonoma County. I mean, that was just an Italian guy whose family said, come on over. And he brought some sticks with him. As I said, we as humans like to bring our alcohol with us. And so, I mean, they didn't know what they were. They may have said, okay, I have this favorite vine. It makes the best grapes, or it's the most cold hardy, or these grapes taste riper to me on this vine than these other vines. Okay, let's take sticks from this vine and we'll replant it.
Yeah, I've heard those stories being done in Napa. That is kind of fascinating. Rick, we're going to jump back to you again. I have a question. What's your view on the current mutations that we're hearing about regarding COVID? And I'm sure that is more than a 2-minute answer, but what would you have to say generally?
I would say I'm utterly unqualified to discuss that, but evolution dictates that there will always be mutations, and we know viruses are always going to have mutations. So the fact that there are changes is not in and of itself concerning, but time will tell whether those mutations have any consequence to how this virus is functioning. So that's really difficult to tell, to look at a DNA sequence and know the consequence. And unfortunately, we just have to wait and see.
Well, maybe this is a tangent off of that. Did fighting viruses throughout our time make our immune systems more resilient against bacteria now?
Yeah, it's a cool question. So if you're exposed to that over and over and over again, your immune system figures out ways to adapt to that very quickly, right? We call this adaptability. The innate immune system, the antibody mediated system that vaccines elicit, is a very nice example of that, right? So there are all these genes called innate immune factors, and they just sit there waiting for viruses or bacteria to come, and they attack them and try to kill them. But the adaptive immune system waits until it sees something and then makes a response specifically directed at that pathogen that is there. So it's this really nice complementation of being primed for things that might have been seen in the past and being able to adapt to things that we haven't seen before.
That's interesting. And then there's a question, I believe this is going back for Denise and Brett. Are there bad clones that have to be destroyed?
There are bad clones that make bad wine. They don't have to be destroyed, but they shouldn't be planted. Fortunately, the economics will take care of that when you've got a vineyard that either underproduces or makes wine that doesn't suit what the markets demand.
Or they get viruses.
They get they get pulled out and they get either replanted or planted with something else.
Grafted over.
Or they might get blueberries or apple trees or cherries. So yeah, there are bad clones out there, yeah. Or, even more, they're not bad clones, they're just better. Because in the wine business, if you're standing still, you're falling behind. You have to keep getting better, keep making better wine, learning, which is why we're here today. We're learning. This is how we get better at
Right. Well, I think you've done pretty good. I think they're good so far. So don't know what better would look like because I'm enjoying them.
Cindy, Laura Rudderman had a question.
Yes, go ahead, Becky.
Laura, go ahead. Thanks. I had to find my mute button. Rick, you may say you are also unqualified to answer this, but so this, just before I came here, there was a thing on the news about how the Bristol variant has been found in the United States for the first time, and it made me think, should I be super pissed that people are traveling? Because do you only get a new mutation in the United States for the first time because someone went somewhere, got it, and brought it back? Or are the viruses everywhere, and are viruses everywhere always mutating, and so it is mathematically possible or likely that this variant sprung up out of sort of sui generis, out of mutations in two different countries at once? Does that make sense?
Yeah, absolutely. I mean, there's a cool evolutionary question there, which is that I think the coronavirus genome is about 30,000 base pair. I can't remember exactly. But like that's one nucleotide change that when we talk about a variant, it's often one single change that sometimes may have a consequence for how that virus replicates or functions, may not. But the probability, as you say, that would happen 2 times independently is incredibly unlikely, right? So
okay, so this is monkeys and typewriters and Shakespeare.
Almost certainly, right? It has to be past
somebody. So I should be pissed, is what you're telling me. Somebody went somewhere.
I'm not going to tell you how to feel, but almost certainly it's because of travel. But another cool angle to that is you might see the same function. So maybe there are variants that become more transmissible that might occur via different changes in the genome, but they have this convergence on the same functional consequence of that mutation. We call that convergent evolution, where the same consequence, the same outcome happens from these independent mutations. And we might be seeing that with some of these variants, but yeah, when we talk about one particular variant, that is a single origin, almost certainly.
Could I ask Rick a follow-up on that, just dead on?
Yeah, please, Stefan.
I think, yeah. Sorry I was late, by the way. I had to get off a work call to go get my wine. The note that you had asked, Laura, I feel like I just read something fairly recently about mutations where they had said, and Rick, this is a question to you, this is not an authoritative statement for me by any stretch, that one patient in the US, like around June or July of last year, had 14 smaller variants inside, most of which were not negative, they were just variants. So like you do see that. So to your question, Laura, I think the way I took that article was that that person didn't go anywhere. It's just that that's what it did inside their own system while it was replicating, while it was doing it. You do get transmission replication also from different stuff, but that one person got it, and just as the host, then he was now transmissible with 14 different types. So my note was really to Rick there is, if that's true, I think you could be a little less annoyed with someone because it's not necessarily like you could just become a host for that kind of system anyway.
Yes, and if there was a new variant that was brand new that was found in the US first, that would be to your point. But this was a variant that was, it was called the Bristol variant because it was first found in Bristol in England, and now it's here. So that means somebody went somewhere. And I now
agree with you. I think you should be mad. I was trying to help not be mad. I agree now.
Yeah, so there will be variation that always happens, right? Because there are errors that happen in copying things. Just like when you copy a note or you try to type a sentence, there's going to be errors. So that happens in the replication of this virus in every single individual. But when you see the exact same change happen, it's almost certainly a linked event, right? And if that change made that virus more transmissible, logically it makes sense that would make that thing travel further. So
we have time for one more question, and I see that the Brenda Collins party has their hand raised. So I'm not sure who there is asking the question, but somebody come forward. You guys are muted. Hi, Glenn.
Hi, Cindy.
That's all right.
Hey, our question is for Brett, and it's about winemaking. You talked about blending different clones in this example, but we're curious how many sort of test batches you have to make. What goes into making a blend? How long does it take? And how many tries do you have to make before you decide what you're going to bottle and sell?
And can we come to your house and help you test?
It's actually, yeah, I know we're not digging a ditch or shoveling snow, but it's actually harder than you think. We start out initially around this time of year, just reviewing all the wines from last year and the previous year. In essence, we're just building a database, just tasting through what works, what doesn't, what do we think.
And by the barrels too.
By barrels. And one thing in winemaking, clones to me is very important. Other winemakers don't think so, but this is not a monocausal thing in wine. Wine's very multivariant. There's 1,000 little steps that go into wine. But from there, we'll go through probably in March and April and start doing test blends. Sometimes it's really easy. The wines are very expressive. You can say we line up 4 100 ml beakers. We have our various percentages. We taste through blind. The key for us is to taste wines. We know what it is, but we don't know what the percentages are. And so that way you don't want your own personal biases, because sometimes I want a certain thing to taste better than what it does.
You pay more money for something.
Yeah, it's like, okay, this vineyard that costs $10,000 an acre, I want it to taste good. And if it doesn't, well, oops. And so from there, we'll further refine till probably in June, we'll actually kind of say, okay, what's the blend going to be? Some wines are very easy to blend. Some wines are incredibly hard. They're just neither not evocative. And the problem is I'm trying to predict on a wine that maybe is 8 months old, what's this wine going to taste like 10 years from now? That's my job.
And how much can we sell? Like maybe we don't want 300 cases of a wine. Maybe we only want 250, or it depends. So there's that variable too. We've
had wines where we probably have done 15 different iterations and blends over 3 or 4 different tasting sessions. Or we'll go through 7 or 8 iterations, say this isn't working. And it can be single percentage points of, say, Petit Verdot in a Bordeaux blend. It can really vary that much, where it really just tips the wine over to a place either we want it to go or we don't want it to go. So when I say, it beats shoveling snow twice a day for 5 days in a row, like I've had to here, both at home and at the winery. But it is hard. You have to focus. You have to do it when your palate is fresh, which is generally around between 9 and 11 o'clock in the morning. And it may take several, times of doing it. And as I said, this isn't really a bad job. It's one of the perks of me doing what I do for a living.
Yeah, thank you. Yeah, I've always been curious about that. So thank you very much.
And so, you come by, you just got to be able to taste a lot of wine and not be drunk. That's kind of the key.
Well, stay nearby.
Yeah.
Thank you, everybody, for your great questions. And I know that we didn't get to everybody. I do just want to add that Rick's talk will be made available so that we can, if you're interested to hear that again, hear what he had to say, we'll make that available again. And if you have ongoing questions, please, I encourage you, reach out to PNRI. Rick's information is available on the website. And please also reach out to Denise and Brett at Isenhower Winery. And we would love to continue the conversation with you. And in closing, I would like to just give an appreciation. I know we have a lot of board members tonight with us this evening on our call. And I would like to introduce to you Tom Hayward, our board president, just to say a few words and to close out the evening for us. Tom?
Thank you, Cindy. I am the chair of the board of PNRI. I'm very proud to be in that position. We're very fortunate as board members to be able to listen to Rick and to Michael, who I see is in attendance tonight, one of the other investigators. And I listen to Rick talk, and I always think of my kids on Christmas Eve. He's so enthusiastic about subjects that are way over our heads. And that makes we're mere mortals compared to him. It's exciting to listen to him talk, and PNRI is asked questions which are different than other scientific organizations. And we do it through, they do it, I should say, through collaboration. It's not in competition with your other scientists, it's through collaboration with those scientists who are down the hall or on the floor above you. It's an important concept. I want to thank Isenhower Cellars, Denise and Brett, really interesting. I knew nothing about wine at all. Cindy Haba, my word, Cindy, the work you put into these things and momentum coaching, thank you very much. I'll just raise a toast. Just to science in general and the unique questions that PNRI asks and how it's going to affect all of our futures. And I'll just raise a toast to our futures in general in these troubled times. Thanks for attending tonight.
Excellent. Thank you, Tom. And this concludes our first Science and Wine Club series. Again, we're so thankful that you were all here. We hope you enjoyed it. We hope that you will join us next time. So when we say it's a series, we mean that there's more to come. And so you can sign up for the next ones. You can bring more friends. There will be more interesting science. There will be more lovely wines to drink. And so we look forward to having you join us again the next one. So I hope you continue to enjoy your glasses tonight. Brett and Denise, thank you. They're delicious. Rick, you've given me just lots to think about. And I appreciate seeing all of you this evening. Have a good evening.